helps distill down the science, make it a little bit more understandable.
Sunday. She was hoping to get an epidural today. So.
Oh, I hope that works. Enough for that. Yeah, exactly.
ALCS, American Lung.
It's your lung cancer foundation. Yep. So that's like if you're comfortable while you're standing. But if there's nothing, take it down.
Okay, I'll stay standing until then.
all can make it? 1, 2, 3. There we go. My name's Bridget. I'm with Penn TV. I'm in charge of your production, the video production that's going on here. That's why we have all the video cameras.
Everybody has signed the okay.
Just super quick. If you have anything to say, whether you're introducing yourself or a question, please make sure we get you a microphone. There's several scattered throughout the room. We're live streaming this. See, this is viewed. Some of you have watched it online, and we're viewed all over the world, which is just crazy amazing. So they can't hear your questions if you don't have a microphone. And after we're all done, James is asked, James, he's doing kind of some pretty stuff for the living room to use for promotional. They want to take a nice group picture with everybody in front of the Hope sign. So don't run out the door. We want everybody in the picture. And we are going to start in about 4 minutes. So get your drinks, oh, that's right. If you have a cell phone, please put on a mode that won't make any noise. And thank you for coming.
Unless you're a doctor.
Yeah, I think most people, did you all sign sign in before you came in the door? There was a clipboard here saying that you agreed to be on camera and all that good stuff because we need you to sign the release for that. I saw one of your folks standing there. Yeah.
I'm going to be passing this around.
You're going to be on TV too.
Look at it, and if you're interested, sign it, please.
What is it for, Sally?
A luncheon. I'll talk about it.
I got you on the agenda.
When, Sally? October 10th.
Yeah, that sounds good to me. I had to miss the last one.
What?
How are you?
I'm good. Are you?
Okay.
Good.
Go Rash.
Let's roll cameras in.
No rash?
Go Rash.
I don't either, but
No, go Rash. I mean, it's still
still going.
alive and well. And
Yeah. Well, I went down on the 100 milligrams. I'm not having rash. How do we get there? The damage was done to my head before my hair is gone. Also, take a wig.
Somewhere. I know.
Bring me a bottle of water.
Sally's here. All right, 4 has a red light. So on the long
Getting back to a normal life. With your little one, it's not so normal, huh?
Well, no, here's you.
I know about you because you're in a home.
He knows about it.
I'm only climbing up the beginning. So little by little, you figure it out.
Thank you, but I just.
What's the medical record?
Yeah, we got plenty of room.
John, you are okay.
Good evening, everyone. Welcome to tonight's Lung Cancer Living Room. Thank you all for coming, everyone in the room. Those of you online who have tuned in, thank you, thank you. Those of you who will come back and watch post-live, either the full 2 hours or the edited version, we welcome you. For those of you who don't know me, I am Danielle Hicks. I am number one, Bonnie's daughter, the oldest daughter. And the other one may introduce herself a little bit later. But my role here at the foundation is to oversee patient services and program education. So everything that has to do with the patient touch and information and working with these wonderful docs that we bring to you here is my area of responsibility. So welcome, welcome, welcome. Tonight, as you guys know, and you have seen in whether it was through social media or direct mail from us, Dr. Heather Wakelee from Stanford is here. A lot of you in the room I know her. I know some of you in the room are her patients. So I don't have to go too deep into how amazing a not only human being she is, but a lung cancer physician. She really does a great job. And she is loved by many. So I'm going to let her do her actual introduction when it comes time, because what I've found is I could sit here and read bios from a paper, but it comes across way more personal when the people we have in the room are talking about who they are and why they chose lung cancer as an area of specialty for them. So we're going to do the same thing we do normally tonight. We have a big crowd, so I'm going to ask people to kind of keep it short and sweet. Who you are, why you're here. If you're a patient, please identify. Give us a little bit of history. If you're a loved one or a caregiver, we want to hear that about you too. And if you came from far, we want to hear where you came from. Same goes for people online. Michelle's moderating the YouTube live stream, and David LeDuc, our executive director, is moderating the Facebook live stream. So if you want to give a shout out and let us know who you are and where you're watching from, please do. With that, I'm going to ask you to do a quick introduction, and then we'll go to Dawn. Who are you?
I'm Danielle and Andrea's mother, and Jared's mother. I do have a son who doesn't get a whole lot of credit, occasionally. But I would really like to say something about, my personal relationship with Dr. Wakelee, which has been, 10 plus years now. Aside from being an amazing physician, she's also an even more amazing researcher, and she's also a mom. But she is 1,000% patient-centric. To her, each patient is very unique and individual, and she treats her patients that way. And if everyone in the world did that, what a wonderful world it would be. So I'm just overjoyed that she's here again. She keeps saying okay and keeps coming back, which is awesome. I'm a 14-year lung cancer survivor and started this foundation in 2006 because of all of the injustice in lung cancer. The survival rate then was only, I think, 14%. Tarceva was in clinical trial at the time, and that was the only drug outside of chemotherapy. So it was the beginning of a new era, and look where we are now. We've had approval on 3 new, not necessarily new drugs, but 3 new uses for those drugs just in the last 7 days. And big shout out to the FDA for fast tracking all of these amazing therapies for all of us. So I'm not going to say anything more other than that there's great hope out there today.
And one of the things I want to tag on, just because I can't not always tag on to something.
Exactly.
Is that since in 2006, when she and my dad decided that they wanted to start this, obviously with full family support, they were going to have a little golf tournament and maybe a little gala, and she had surgery up at UCSF, and Dr. David Jablon, she was very grateful. He had a research lab up there, and she was like, oh, I'm just going to give all my money to David's lab, and that's going to fix things. And it didn't take a whole lot of time before we realized that was going to fix absolutely
We did give him $1 million.
Nothing. Yeah. Yeah. But what I will say, and one of the things that I'm most proud of, is in a very short amount of time, we have raised right around $40 million, and roughly 80% of which goes right back out the door to support our mission. So, yeah. So I'm really proud of that. We have an amazing team of people, some in the room tonight who are able to be here, who I will ask to introduce themselves very quickly and say what they do. So we definitely don't do it alone, but I just wanted to say that. So, Don.
Thanks, Danielle. My name is Don Stranathan. I'm a stage 4 lung cancer survivor. I want to welcome everyone and everybody online. I know I have a number of people that have tuned in online, so welcome. And I was diagnosed with stage 4 lung cancer in June of 2009. And like Bonnie had mentioned, back then there wasn't a lot of targeted therapies. So, I went through chemotherapy, a couple different chemotherapies, one for my lung, one for my liver. And then I was handed Tarceva, and they said, well, try this. And it worked. It worked for over 6 years and kept me stable. And then in 2015, I had a new tumor in my left lung. And I decided to go ahead and have next generation sequencing done on it, which wasn't even available when I was diagnosed, and found out I was an EGFR. So I reached out for a second opinion, was suggested immunotherapy, which wasn't also available in 2009. And since then, I've been on a number of different immunotherapies. I've had some minor progression. I'm doing extremely well right now on a new triplet of Keytruda, Limta, Carbo, the Keynote 189 trial. And after only 4 infusions, I'm on my 9th infusion, but after 4 infusions, I had over 50% shrinkage on all of my tumors, even the ones that I've had thought were scar tissue. So anyway, welcome. There, like Bonnie said, there is hope. So thank you.
Thanks, Don.
I'd like to, please, thank you, Don. I'd like to say one thing. I don't know if everybody in the room or everybody online knows that the living room here has reached 144 countries. So the purpose of the cameras in the room are the fact that we're live streaming on YouTube, and we're also doing FaceTime Live right now. So this is happening all over the world, and this is to educate patients, and in some cases, physicians in other parts of the world that don't get the continuing medical education that's offered here in the United States. So just a little anecdotal information about why all of this chaos is happening.
Sure. Real quick, Jason, glad to be here. I'm a patient advocate, and so I'm just here to learn and listen. So thank you.
Awesome.
Thanks, Jason.
Use the yellow one right there.
I'm Joanne. I'm local from Belmont. I am a 2-year survivor, or we could call it 3, because I was misdiagnosed with a cough for the first full year. So I don't know how we count that.
We'll go with 3.
I'm surviving 3. Okay. I am EGFR 19, exon 19. Have been on Tarceva for the 2 years. Quite successfully. My tumor started at 5 centimeters, and it went to under 2 in the first 6 months, and that's where things have stayed, no progression. I have suffered some side effects. In fact, one that I don't know, hadn't heard of anybody else having, was scabs on the scalp, which has resulted in loss of hair. So I have a lovely wig, and I have scarves and hats, and I'm very happy. My dose was reduced to 100 milligrams from 150 2 months ago, which has, the damage to my scalp was done, but it has stopped or cut back other side effects and other rash and so forth. I had a scan yesterday at the 2-month mark on the 100, but don't have the results yet. I'm also in a blood biopsy trial that I should have, for progression, that I should have the results of, the first results on Thursday, I think.
Thank you.
And this is my caregiver, but we were camping and he took a trip down the mountain.
Oh,
and
my
so
goodness.
I'm the caregiver at the moment, so excuse me.
So another patient.
No.
And thank you, Nurse Ratchet.
Hi, I'm Sally. I put myself into an early lung cancer detection study to see if low-dose CAT scans would pick up lung cancer before regular X-ray did. I was at high risk, and you had to be at high risk in order to get into the study. And 5 years into the study, the little nodules they found, one had grown. And because I was found so early, I had surgery. I had my lower right lobe removed. Knock on wood, I've been at NED since, and I still have other nodules, but they watched that, and it's been a little over 9 years. So I'm very fortunate.
Thanks, Sally.
My name is Tina, and I was diagnosed in 2010, so I'm going on 8 years pretty soon.
Yeah,
So
yeah.
I'm happy to be here, obviously. I was EGFR positive, but I did do chemo and radiation, and then I went on Tarceva after a clear scan, and that was 7 years ago that I've been taking Tarceva with clean scans. So I think I'm, what is it, outlier or something?
You're an outlier.
Anomaly?
Yes, definitely. For many more reasons than just that. Yes.
I'm Rick, Tina's husband, and supposed caregiver. But like Don says, I mean, I haven't fallen down a mountain, but she takes care of me more than I take care of her.
Hi, I'm Evie Schiffman, world's luckiest person. I had early detection as a result of reading an article in a newspaper about going to get a heart CT scan that wasn't looking for lung cancer, but they discovered it. So now I'm a 2-month survivor. And I had surgery in June, and I'm NED, and I am very, very, very fortunate. And prior to being a cancer patient, I was caregiver for my husband, Neil, who passed in 2015. Unfortunately, he wasn't discovered until stage 4. And I would just like to say Dr. Wakelee is our family oncologist. The only one in my family that she hasn't treated is the dog, Cubby. Thank you.
I know who you are.
I know, and I know what your
You're
story
his
is, too.
sister.
My reason for being here is a little different. Before Bonnie has a chance to tell you, I guess I will, because she didn't last time. My brother, Dr. Fred Marcus, was her physician before he passed away. And so once that happened, I didn't know a lot about lung cancer, and I really questioned a lot about why, how, and my brother just told me it happened. So I decided that I watched Bonnie build this from the ground up, and my brother worked here, so yeah, that's him in the
He's back there behind the flowers.
Behind the flowers, yeah.
And I came to Bonnie, and I literally cried on her shoulder, like how this happened. So she set me straight and made me feel much better about it. So ever since then, I've kind of been proactive in discovering any information that I can to find out kind of what he went through. And that's kind of my story.
Thanks, Debbie.
Hi, I'm Kathy Vonderporten, and I've lived here for 30 years. And my mother is newly diagnosed last week. And so we're grateful for this opportunity and resource. So I'm the oldest of her 6 children and one of her caregivers. So we're happy to be here. I'll let her tell you the story.
Thank you. I'm Chris Coons. I live in Lafayette, California, here across the bay. And I'm ever so grateful for my daughter, who began some serious research on this subject and discovered this location as a resident of San Carlos many, many years. And I happen to be legally blind about 11 years ago, and am awaiting this information. I'm on a journey of research and education, and I'm very happy to be able to be here today. Thanks to Kathy.
Thanks, Chris. Well, we're happy you are
For
here
Bonnie.
today. I'm going to come give you a hug pretty soon.
Yes, I'm okay. We've got one here. My name is Dave, and I was diagnosed in March of last year, 2017, EGFR. And so I've been on Tarceva since then, and it'll be a year and a half next month. I just came from the oncologist a couple hours ago. So today is like cancer day. And my oncologist said, make sure you say hello to Dr. Wakelee, because she did a little training under Dr. Wakelee. So, and so, by the way, so did our guest speaker from a couple months ago, Dr. Blakely. I guess he speaks highly. You bring the whole party. Yeah, it's great. We've got her.
She has a posse.
I'm waiting on some. I've got some fairly inconclusive results on a recent biopsy of a lymph node, and so we're going to do another PET scan in a couple weeks, maybe 3 or 4 weeks, and then we'll have more information then. So thanks.
Okay.
Thanks, Dave.
I'm just the wife, but I'm not really his caregiver. I am more his communications manager, and I send out emails after every time we have something new. They go to about 300 people.
Right.
And I will only say that the last one we sent out said, well, we're in limbo because we really don't know what's going on. And after today's appointment, it was like, well, we're still in limbo. So sometimes this happens, and I guess we say the good news is that like they're not saying, oh my God, you're dying, we got to go into surgery now. So we'll take what we got today.
Okay.
Thanks, Barb.
Okay.
Oh, hi, I'm a caregiver to my beautiful food scientist here who's doing a lot of research, and I'll let her talk. Thank you.
I'm Anna Calloway, EGFR, stage 4. I was diagnosed February 2016 with adenocarcinoma. And currently I'm on therapy of Tagrisso and chemotherapy of Alimta and Carbo. I've had 3 infusions, and so far things are looking okay. My treatment is at Stanford with Dr. Millie Das, and I'm very, very grateful. It's a great place, and I'm happy to be here today.
Thanks, Annie.
Hi, my name is Maria. I was diagnosed at 43 years old and stage 4. And I had surgery. I had my right lung removed. And then I found out I have metastasis in my brain. And then since February, and I had cyberknife in my brain in February 2016. And then in February 2016, I started Tarceva, and so far it's been working. I'm fine. I'm alive, so I'm 46 now.
And you look awesome. You look absolutely awesome.
My name is Deborah Frank, and about 10 years ago, I was diagnosed for the first time with lung cancer and met Bonnie, and I actually met you, Dr. Wakelee. I was one of your patients, but you told me we didn't need to do chemo. And I had surgery and had my right upper lobe removed. I had the EGFR exon 20 mutation, and I can't believe I know that. And after my lobectomy, I didn't need chemo or radiation or anything, and I was sailing along fine, going, okay, I can do this. And then 18 months ago, I was diagnosed with a new primary, and I had radiation at Stanford, and last week they told me that I'm net again. So I told them I'd see them again in another 9 years, and they said, no, 6 months. So that's my story.
So NED, NED, for any of the newbies in the room means no evidence of disease.
My name's Linda Salzer, and I'm making notes of things like that because I'm here on behalf of my sister Lori, who's watching right now from Lake Oswego, Oregon, who was diagnosed last June with stage 4, and I love you.
Oh.
Hi, Lori.
That's a sister. That's a sister.
Hi, I'm Fran Costello. I'm actually the president of the Patient Advocate Foundation. I've come from Virginia this morning on a very, very early flight, so I think I'm working on about 18 hours now to learn more about this foundation and the work that it does on behalf of all of you. My family actually started the Patient Advocate Foundation in 1996, and our mission is to serve chronically and critically ill people who are encountering access and affordability issues, so insurance problems, i.e., or cost of living challenges that you might face, or disability applications that you need to make. So the very practical issues that people face when they're suddenly or for a long period of time dealing with illness. So thanks for having us.
They're amazing.
So are you. And I'm Alan Richardson. I'm the Executive Vice President of Strategic Patient Solutions at the Patient Advocate Foundation. I've been up the same amount of time as her, and everything that she said about the work that we do, it's just a pleasure to be here, so thank you.
My name is Ron Fong. I'm here tonight as a patient advocate, and also because I heard that Sally brought coffee crunch cake. We've been having a taste-off between a Chinatown version and a Peninsula version, so we'll see tonight if that's really good. And for the walk that's coming up in September, someone has challenged me to wear a red dress and run around the lake in a red dress.
Yes.
Yes. Absolutely.
That does not swing the way I normally go, but but Joan loved the color red, so I'm contemplating it, and we'll see.
Okay, all right, okay. I think he's going to do it.
My name is Fred Palmer, and I think it was about 5 years to the day I had a chest x-ray that showed a mass on my right lung, subsequently diagnosed as lung cancer. And I'm currently on immunotherapy, and it seems to be managing it quite well. So I'm still here after 5 years now.
Hi, I'm Nancy Waringa. I live a long ways away. I live in San Carlos. Anyway, this is my first time here. I'm grateful to be here. Thanks to Don. I have small cell cancer, and I started out last year with being in the hospital for 3 months with a low salt issue. And I thought I was going to die from the low salt. And my last day in the hospital where they couldn't do anything, suddenly one of the interns decided to do an extra test, and that's when they found cancer. Did the biopsy, and then they said small cell, and I knew it wasn't good because everyone's faces dropped. But anyway, that was November. I'm still here, and thanks to you, I'm learning tons. So thank you very much.
Hi, my name is Nancy Miller, and I came from Fresno. I am with Novartis, and we have an oncology department, and I sell a product for non-small cell lung cancer, and I really wanted to hear whatever Dr. Wakelee wanted to hear. I didn't anticipate getting to hear all your wonderful stories. Thank you so much. It makes me feel like I'm doing something that's helping people in a really big way.
So my name's Jim Cooper. I live over in the East Bay. I as well work for Novartis, but the reason why I work for Novartis is that my wife had breast cancer. So she is a 6-year warrior, but I figured this is one way for me to immerse myself in the oncology community by actually working in it, getting to meet the patients, and hearing all your wonderful stories, because it makes me work that much harder for all of you. So thank you for allowing us to be here.
Hello, my name's Nellie. I'm here to help support my daughter, Rebecca, down there.
Hi, I'm Chloe. I'm here with my mom, and I am one of her part-time caregivers, along with my stepdad.
Hi, my name's Wayne. I'm a caregiver for my wife, Rebecca. She was diagnosed in November 2017 with adenocarcinoma stage 3B. We started chemoradiation and then immunotherapy, and we waited 9 months for our genetic testing results. And we were kind of disappointed because we ended up with garden variety KRAS. So she started chemotherapy last Wednesday, carboplatin and Alimta, and also to our surprise, she had a 2-millimeter little nodule in her brain that 3 doctors that were just assists, and that turned out to be a brain metastasis. So next week she's going to do the CyberKnife, and we're hopeful that that's going to turn out good. But yeah, and this is kind of new, and we're just waiting for the next step to happen and hoping for some good results.
Thank you.
Hi, my name is Breeze Keeley.
It's my first time today, and I'm here with my family. This is my partner, Peter, and my sister, Marley. I'm a physician at UCSF, and our mom died of stage 4 lung cancer 4 months ago. She was diagnosed a week before she died. She was a very special and amazing woman, and was not someone you would expect to develop stage 4 lung cancer. We were all with her at the time, and she was getting ready to move out here with us and start treatment, and unfortunately the tumor embolized to her brain, and she died of complications of that shortly thereafter, and it was very peaceful. So we learned about the Bonnie Adario Foundation after her death and started a memorial fund in her name, and it's been really meaningful, and it's been a wonderful organization, so we've been very happy to be able to give back and learn more. We're happy to be here today. Thanks.
I'm Ingrid. I come with my friend, and actually I was in the insurance industry for a decade, and I want to help my clients more besides, the monetary way. Okay.
My name is Evangeline, and I'm glad to be here. I was encouraged by my social worker and case manager at Stanford to attend this support group because she said Dr. Wakelee will be here, and she's a colleague of my medical thoracic oncologist, who is Dr. Neil. And I've never had any experience joining a support group, so I said, okay, I'll come, and this is my first time. I was diagnosed March of last year of NSCLC, non-small cell lung cancer adenocarcinoma, and it was incidental diagnosis because I was accepted at Lucille Packard Children's Hospital to be certified nurse assistant because my dream is to take care of children, and they suggest everybody before we start work, I already had my badge and my orientation, but I was required to have chest x-ray to make sure that I passed the TB test. So I passed the TB test, but they found a mass on my upper left lobe lung. But I never had, so my primary care physician was so surprised and shocked, as well as myself, because in December 2016, I had excellent results of blood work. My doctor said, you're perfectly fine, you're healthy, except you have to exercise because you're overweight. So I started joining jazzercise. So February, I had this chest x-ray, and they found this mass, and my primary care physician said we have to make it fast. So every week I had procedures like CT scan, they found out it's cancer, and then a biopsy, so they found out it's malignant, and then she referred me to Stanford Cancer Center and referred me to oncologist, and I was scheduled to have my surgery April 27th after the PET scan, but day before the surgery, they forgot to do a test on my head. So day before surgery, I had an MRI, and 9 o'clock in the evening before my surgery day, they called me to cancer. They said they're going to cancel my surgery because they found out I have 2 tumors, one in my right brain and one on the left bone of my skull. So April 27th, they categorized me as stage 4. And May 17th, I had my cyberknife radiation on my right brain. And I started my Tarceva, the chemo on Tarceva, May 22. After 7 weeks, the tumor on my left lung has reduced by 1 centimeter. It was originally 3.6, and now it's 2.4. It's as big as a plum. Now it has been reduced. By the grace of God, August 6, I had my whole body PET scan, and August 7, I have an MRI of my head, and my radiation oncologist and my medical oncologist says it's all dormant. So I'm thankful for that news. Thank you.
Hi, my name is Tom. I'm here for my partner, Melissa Scott, who had been diagnosed in January of 17 with EFGR stage 4 lung cancer, and she went through 3 loads of chemo. Then she started on Tarceva, came up, and it worked well for 14 months. After that, it was no evidence of disease. Then they checked her again about 3 weeks later. She ended up showing some signs, and they started on her Xalkori. So it mutated to the ALK mutation, which is a very rare, what I understand, like a 3% thing. I don't know numbers too much on that. We were living in Wyoming at the time. We moved to, she wanted to come home back to Calaveras County, which is up in the foothills. Angels Camp, Murphy's is where we moved home to be around her family. She thought she had it whipped, and she was the strongest person I've ever met in my life. And I'm grateful to know her. I love her. But she just passed 2 weeks ago, and she gave it a hell of a battle. Sorry about my language, you guys. I didn't mean that. And she promised that we needed to be here, bring some flowers for all you guys. Bless all of you, and that everybody. And we were online every 3 weeks. We were here. That big word on that wall there is what she lived for. So everybody has hope, and there's just bless all of you so much. God bless all of you.
Hi, my name is Devin. I am a patient advocate with Biodesics. Coming from Colorado, we're just really happy to be here. Thank you for sharing your stories.
Hi, my name's Abby Wright. I'm a colleague of Devin's and Linda behind me, also with Biodesics, also a patient advocate. Biodesics, we do genomic and proteomic testing to help avoid some of the longer issues of getting test results back to patients. So we try to do that and do it through blood, so it's less invasive. And my particular job at Biodesics is working with the health plans and our customer care team to ensure that any tests that we run are covered by insurance and are not paid out of pocket for patients. Thank you for having us here today.
Thank you.
Hi, my name is Val, and I'm here with my wife, Galina. She's been diagnosed with stage 4 non-small lung cancer last October. Fortunately, she has EGFR mutation. And another fortunate thing that we're under care of amazing Dr. Wakelee. And so far, she's taking Tagrisso, and she's doing okay, works.
Hi, I'm Mike Jones. This is my first time here. I'm in the industry. I work for a company called Caris Life Sciences. We do genomic profiling for lung cancer and various tumors, and worked a little bit with Dr. Wakelee. And this is a treat for me because I spend my whole day in front of medical oncologists, and I'm looking forward to learning from all you tonight.
Yeah, I'm going to ask, so I see some staff members out in the hallway and there's some back here. If you guys just introduce yourselves really quick so they know who from our amazing staff is here. And of course, it's not everybody, but I see Sandra and Leah back here, and Andrea and Nicole and Katie.
Come on in.
Just really quick, sorry.
I wasn't expecting that one. I'm Andrea Parks. I'm Associate Executive Director of Development and Partnerships here at the Foundation. So I work with companies like Biodesics, Patient Advocate Foundation, Novartis, And I'll talk to you in a second, Mr. Karras over there. He's been non-existent. Anyways, I'm also Bonnie's daughter and Danielle's sister here.
The youngest daughter. She
The
always
youngest.
points out. Yeah. Good.
I had to stop doing that when I had to start wearing readers. So Danielle kept on telling me it was going to creep up on me. But anyways, like Devin was saying, just hearing everybody's stories here is it's why we do what we do. And we're here 24/7 for all of you guys all the time. And it was so nice to see Deb. Okay, so Deb found out about the foundation when my sister and I had a shop. And we used to sell these shirts that said a breath away. And then on the back was the Lung Cancer Foundation logo. And that's how she found out about us. And
Evie, too.
Was it Evie, too?
Yeah.
Oh, I didn't even know
Yeah.
that. But
Yeah.
anyways, so it's wonderful to see you here.
Who?
I couldn't believe it when she was here tonight.
Oh, yeah.
Hi, my name's Katie Morello. It's my second day on the job, and I've known the family for at least 25 years, though, so I feel so fortunate to be a part of this wonderful community and look forward to learning more.
Hi, good evening, everyone. My name is Leah Fine, and I'm the director of our Centers of Excellence program, which is a network of community hospitals all across the country that are focused on best practices in lung cancer care, including multidisciplinary teams, molecular testing, and basically the full cancer care continuum from early stage screening and diagnosis all the way through late stage care. And very excited to be part of this program and to building out a network of community hospitals so that, you know, in places like Wyoming and Texas and all across the country, patients have access to the best possible care. If they can't make it between Stanford and UCSF, they have access to amazing hospitals and care teams all across the country.
Thanks, Leah.
Hi, I'm Sandra Shaw, and I'm the director of the Patient Registry. It was a pleasure to meet some of you tonight who've joined the registry and are wearing the I Join buttons. For those of you that haven't joined the registry, we'd love for you to join and donate your data. It's a place where the community can come together, and really the power is in the patient and providing that data. And researchers can come in and look at the data in aggregate and do studies and propose research. So we'd love to have you join the registry, and I'd love to put one of those buttons on you next time I see you. And with that, I'm going to hand this over to you.
Hey, everybody. I'm Amy Moore. I'm the Director of Science and Research here. So you may have seen me on the panel recently up there. And then, you know, really what I can do for you is to help translate some of the content that you may hear tonight. Some of these words might be big and scary and hard to understand. So as you listen and then, you know, kind of contemplate what you heard tonight, feel free to reach out to me. And if I can try to explain things in a simpler way, you know, use me as a resource, and I'll try to help you understand what we're talking about and make those connections. So glad to see everybody.
Tony's right behind you.
Hi, I'm Tony Adaria. I'm the CEO of the Adaria Lung Cancer Medical Institute. We're a consortia of about 26 academic centers and high-end community hospitals in the United States and Europe. And we do some of our own research, some funded by ALCF, some funded by pharma, and some funded through the contributions that that you make either to Alchemy or to ALCF. So we have about 11 studies in process right now in various categories, trying to fill the gaps that some of the big institutions and pharma may not be looking at. So we had some very interesting results, and I'd be happy to talk to anybody about some of the things we've done or some of the things we're doing. Thanks.
Thank you. It wasn't enough to just have one foundation. We had to have two, apparently. Thank you all for sharing your stories. I know I say this all the time, but there's a lot of new people in the room tonight, and I know you touched on it a little bit earlier when we first started the meeting. It can be a little intimidating with all the lights and the cameras, and please sign the waiver before you come in, but the information that you share and the stories that you share are insanely impactful, not only to the patient and caregiver community, but to the physician communities, the industry communities, the other nonprofit communities out there, who, and I think you said it back here, it reminds them why they're doing what they're doing. And it makes them really proud to be doing what they're doing, myself included. So Thank you all so much for sharing that. And with that, I'm going to turn it over to Dr. Wakelee to give us a little bit of information about who she is and why lung cancer matters.
Okay, great. Well, first of all, I wanted to thank you for inviting me to be here. It's always a really wonderful experience to get to talk to so many people at once. I mean, I give a lot of lectures, so that part's not that unique, but the audience isn't always patients and patient advocates. A lot of times it's other physicians. And I want to emphasize, too, it's not just the physicians. There's a whole care team involved in everybody's treatment. So it's the nurses and the nurse practitioners and the pharmacists and the social workers and advocates for a lot of people. And it's really important to emphasize that team. And I like being part of this team, too, and love my opportunities to work with the Adario Foundation. And all of you have come here to be a part of this, but I want to just emphasize how amazing it is what you guys have built over the years. I think we got to know each other pretty early in the days of the foundation. And to step back and think about how many people have been helped, have been touched by this, by knowing that it's very overwhelming. You come with a diagnosis and you meet the healthcare team and you know that they're there as advocates and trying to help you and do what's right for the individual patient. But it's also very helpful to hear that from someone who's not your patient, or not the patient, not your care team, but someone else who's living through the disease or knows a lot of people who have lived through the disease, and to really help to understand it. I think sometimes people are afraid to ask their care team a question they might think is obvious, but it's to be able to ask it to someone else who's going through it, or to know that there's a place to come for support. And that's just so critical. And I can't tell you how many of my own patients I know have come and been really supported here. And then also to emphasize the research aspect of it. Everything that we know to do today, we know to do because other patients who've come before have been willing to be a part of trials and to try out those new treatments, some of which have worked and led to things that you all are talking about now, and some of which didn't. I mean, not everything works, and we don't know that without moving ahead and doing those trials and those studies, and to be able to have ways of doing that research from the ground up in the lab to studies to actually being able to try those things in people. And it's not just the treatments, but it's the understanding the diagnoses, being able to know what's the right treatment because I've done this test and therefore I know this is the right thing for an individual. And so the support that's being done for that is really critical. So I want to emphasize that too. Okay. So then me, I haven't treated everyone here. I wouldn't be able to, right? But I've been involved in treating lung cancer patients in this community for quite some time. So I'm a native Californian, a little further north. I was on the East Coast to do my medical training, but then I came back to Stanford to start with, and for those of you, when you finish medical school, you do a residency, and then you go on to specialty training. And so I came out here to do my internal medicine training, knowing then I wanted to be an oncologist. And so my first day at Stanford, I was on the oncology ward. I showed up an hour late because they gave me the wrong time, but we got over that. And from that time, that oncology ward at Stanford has been a second home to me. Can't tell me how many birthdays I've spent there and other things, but it's been a really important part. And it's that whole group of people. It's the nurses, it's the pharmacists, it's the cleaning staff, the desk staff. I mean, a lot of them have been there a really long time because they see how much their role is in helping people living with cancer. It's not just the physicians. And as I started in oncology, kind of informally in my residency and then formally as an oncologist in fellowship, I realized there was a big need for people focused in lung cancer. So this was a long time ago. And it was before we knew anything about what I'm going to talk about today, all this molecular stuff. We didn't know anything about that. Way before immune therapy. I mean, that's actually, I went into oncology because I remember in college thinking, we should be able to get the immune system to work against cancer. And it took like 2 decades to get to that point, but we gotten there finally. But there was this real need, and I actually ended up needing to go to UC Davis, where Dr. Gandera, I don't know if you have heard of him, but he's a fabulous oncologist and really been dedicated to treating lung cancer for a long time. So I would commute up there once a week for a while to be a part of their lung cancer treatment community and learn from all of the people there. And then was able to bring that back to Stanford. And for a while, it was just me. I was the medical oncologist who did lung cancer. We had great surgeons, great radiation oncologists, a team, but it was just me. And it's because not as many people were interested in treating lung cancer. It was unfortunately seen as well, all we have is chemo. Chemo doesn't work that well. We have surgery if someone's caught early, but nothing after surgery is helpful. So there wasn't as much interest. And that's where I saw a real need to be able to go in and help people who really needed help. More than some of the other, all patients living with cancer need a lot of help, but to me that seemed like an important calling. And now, many years later, it's so exciting that when I have a discussion about treatment, it's not just about chemotherapy, which is still an important part of the conversation, and I'll go into that a little bit, but that we also have to talk about this molecular testing, because that makes a huge impact for treatment for a large percentage of patients. For those of you sitting in the room or listening, it might sound like everybody has EGFR mutant lung cancer. That's not the truth. A lot of people do, and in this area, it's a large percentage of patients. When we have clinic and I see who's on my list for the day, a lot of times it's half the people do have EGFR mutations. But if you go around the country, around the world, it's not always that. And so we'll talk about some of the other things that can happen. And then now all of this immune therapy, which also helps a lot of people. And so it's so exciting to have a lot more things to talk about and to be able to offer to people to try to help. It's also been wonderful that over the years we've been able to grow a pretty big lung cancer program at Stanford. And so you've heard some of the names of some of my colleagues, but now there's Millie Das and Joel Neal and Suki Pada and Kavitha Ramchandran are all medical oncologists just focused in treating lung cancer patients at Stanford and our Palo Alto VA. And so it's great to have a team who are all just really passionate about treating this disease. And then we have 4 amazing thoracic surgeons and a radiation oncology team. And then, like I mentioned, all of the nurse practitioners and nurses and pharmacists, the whole group all working together. And so we have our tumor board every Tuesday, so I just came from that, and that's always nice where we're able to, as a group, really talk about cases where we're not really sure what's going to be the right thing for an individual person, or they maybe need surgery or radiation or both, or chemo or immune therapy. We can all kind of talk together. Okay, so that's me. So I've been here for a long time in this area treating lung cancer, and during that time I've then been involved in being able to train a lot of other medical oncologists who have gotten excited about treating lung cancer and helping people with the disease, including my 4 colleagues I just mentioned, but also colleagues who are now at UCSF and at UC Davis and spreading out around the country, and it's great. It's a really great network. And the medical oncologists who are in academics, a big portion of our job is, of course, taking care of people one-on-one, but it's also trying to educate on a much bigger scale, global scale. And so that involves going to conferences, knowing what's the latest and greatest, being a part of the latest and greatest with the research, but then also informing everybody about that. And so this sort of a forum to have patients come and learn, and it's amazing. We do a lot of this with also healthcare professionals, and I've found that it's really wonderful to be able to travel and to see how people are treating in other places. I actually get to go to Idaho next week to a couple of different cancer centers there, and it's great to see what's being picked up and learned and how practice is changing. And it's also sometimes wow, we need a lot more education to happen to make sure that people know about all of these new things that are happening, because it's happening so fast. It's very hard for someone like me whose only job is to really, really understand lung cancer to keep up with all of it, let alone someone in the community who has to know how to treat breast cancer and colon cancer and prostate cancer and lung cancer, right? It's just really, really, really fast. So it's exciting that it's happening so quickly, and so many things are available for people now, but it's a lot, so that's good. All right, so that's enough about me. So let's talk about today's topic, which is advances in molecular-based therapy. And I go, well, what's molecular-based therapy? Well, I think we're going to back up a little bit as to what is cancer, right? What is cancer? So cancer, it's when a cell, normal cell, changes in some way. It gets a mutation, or maybe a lot of mutations, that allows it to grow in an abnormal way, right? That's what a cancer is. And that happens to all of us all the time, actually. We have an abnormal cell that starts to grow in the wrong way. Usually, our immune system comes in and takes care of it, and we don't end up developing a cancer. But sometimes that doesn't happen. And that single cell then divides and makes more cells, and then they make more cells, and then you've got the tumor itself. And then pretty early on, some of the cells start to break off and get into the bloodstream. And if they change again in a way where it allows them to grow in other parts of the body, then that becomes metastatic. So it can go to the brain, if we've heard from some of the people here today, or the liver, the bone, I mean, anywhere. And when does that happen can vary quite a bit. So some people get a cancer, and even when it's barely visible, one of those cells has already figured out how to grow in the brain, and then it's metastatic. And some people end up with a tumor that's gigantic, and those cells never figure out how to grow anywhere else, and you can just take care of it. So one of the things I try to emphasize is not to focus too much on the size, because the size isn't so critical as much as what is it about that tumor that gives it that advantage? Why is that tumor different than someone else's tumor? Why is that tumor able to grow in different places in a way where it's life-threatening? So that's what we kind of think through. And the reason I'm going into a lot of discussion around that is that it's the differences in each individual cancer that allows us to treat people differently, right? That's that personalized treatment. That's a kind of a buzzword we've all heard a little bit, but it really at the root of that molecular-based treatment. And so when I talk about molecular-based treatment, or when someone does, what we mean is for that particular tumor, we've figured out the gene mutation, and we have a specific way to treat it. Okay, does that make sense to people, right? So in order for a normal cell to become a cancer, there has to be a mutation or many. And what happened about 15 years ago, it was we started to understand, well, what genes are most commonly changed in lung cancer, and we realized that for some of those gene mutations, we can actually treat them, like if they're in specific things. And so I'm going to tell the EGFR story, because that's kind of the beginning of it for lung cancer. And so before we understood that, well, we always knew that in order for a cancer to grow, there had to be mutations, but we didn't understand that, like, well, we could get smart about it until about 15 years ago. So what started off with this EGFR story was scientists realized that if you looked at the cancer, lung cancer, a lot of times there was a protein, the EGFR protein, that levels were really high. And so people said, well, if the protein levels of EGFR are really high in lung cancer cells, and that's different than normal tissue, then maybe we should do something to attack EGFR. And so they went and developed pill drugs that could specifically target EGFR, not the protein, but the DNA part of it. And when they did that and gave it to patients early on, about 10% of people had really dramatic responses, where the tumor melted away within a very short period of time. And those of you who have EGFR mutant lung cancer, you probably remember when you started treatment, for most people you can feel it's actually starting to work pretty quickly initially. But it wasn't happening for everybody, and so that led the scientists to do more work to figure out, well, why is it working for this 10% and not the rest of them? And that's when realized that, well, in the tumors where there was a really good response, for those people, in the tumor itself, there was an EGFR gene mutation, right? So the thing that actually had led to the cancer was that EGFR mutation. And when the EGFR mutation happened, the EGFR protein gets turned on all the time. Normally it's regulated, so it's on, off, all the time. But when it's turned on all the time, it led that cell to have a growth advantage, where it didn't die the way it was supposed to. It just kept going on and on and on and on and on. And so when you then turned off that one particular gene, all of a sudden, the cancer didn't have its main growth factor, and it kind of died. Didn't completely, but at least it slowed down, was able to shrink. And so this was like the first way, the first kind of key to understanding this molecular-based treatment in lung cancer. We knew about this for a few other cancers before, but no one had figured that out for lung cancer. And so they went back and they realized that in all of the tumors where there was, in all the people who had really good responses, if they looked at their tumor and did these mutation testing to see, well, could they find that, they would find these EGFR mutations in almost everybody. And in the people who didn't respond, they didn't have those EGFR mutations. And so it seems obvious now because we know to test, and many of you have been tested and know, but that was like really interesting in news, and it took a while for us to understand how critical that was. So they did a big study where before we knew this information, they realized, well, there's certain kinds of people that when they get lung cancer tended to have these responses. And those people tended to be people who got lung cancer who had never smoked. Not always, but it was a little bit more there. And it actually tended to be a bit more in people who were from Asia or of Asian ancestry, but again, not always, as very obvious in this room. But it was really that never smoking thing that was critical. So they did a study in non-smokers with lung cancer, and they gave them an EGFR drug versus chemo. This is all metastatic. And they saw that the people who had the EGFR drug, that some people really responded and some didn't, but it was still kind of a mismatch of who was what. But then they went back and tested, and they realized that if you could find that EGFR mutation in the tumor, that those were the people who had really good responses. And the ones who didn't have it didn't do very well with those EGFR drugs. And so that started our kind of paradigm shift where we started testing. And if someone was found to have an EGFR mutation when they were first diagnosed with metastatic disease, we knew to start them on an EGFR pill. And that's why so many of you are on erlotinib, which is also called Tarceva. And some of you are on osimertinib, which is also called Tagrisso, right? That we know that if you have an EGFR mutation and you get an EGFR drug, it works better than chemo if you're just getting started on therapy. And so that was a whole new way to think. But we also then stepped back and said, okay, well, that's amazing and wonderful for the people where we can find this EGFR mutation, but it's only about 10% of anybody who gets lung cancer. And obviously it's a higher percentage here, but it's about 10%. What about everybody else? What about the other 90% of people? And so then we started realizing we could look for these specific gene mutations. And then a little bit later, a few years later, we found the ALK gene mutation. So some of you have probably heard about ALK. And then there was one called ROS1, and then there's BRAF, and now there's MET exon 14, and I mean, I could keep going. But basically, what we know to do now is when someone's newly diagnosed with metastatic lung cancer is we have to look at the genetic profile of the tumor. And these are not things that are in the rest of the person. They're not things that are passed down to the kids. This is something that is just unique to the tumor. It's what makes the tumor different than the rest of the body, is that specific gene mutation. Does that make sense? Right? And so that's really the basis of what is molecular-based therapy. It's understanding what is the gene change in the tumor that makes it the tumor, and can we then do something about it? And so some of the things, yes, we can. The ones I mentioned, if we find an EGFR mutation, we have many drugs now that work for EGFR. And if you find ALK, we've got many drugs that work for ALK. And if you've got ROS1, we've got a couple. And in BRAF, we've got some combinations. And MET, there's a couple. And again, the list kind of goes on. And so that's a really, really important thing to know if you have lung cancer is, well, what are the genes in my tumor? But it's not, again, enough to help everybody, because then there are a lot of other mutations that we find. And one of the most common is KRAS. And with KRAS, we don't yet have the right combination that really works especially well for that, though we are just opening a trial at Stanford, so something to know about.
KRAS trial.
And there have been many other trials, and unfortunately, they haven't hit the mark in the same way we have with EGFR or ALK, but we're going to keep trying and trying to understand that. And then there's a huge list of other genes that we know play a big role. One of them that comes up all the time is this P53 or STK11. They're all a bunch where we know that that's a really important mutation that's driving the tumor, but we haven't yet figured out how to go after that and change what we're able to do to help someone with it, okay? And so that's unfortunate, but of course the story is more complicated than that, right? So some people with a tumor, we can find a single gene that's really the key player, and when we do that, we can give medications some of the time. But for other people who get lung cancer, and as time goes on, all the tumors develop a lot of different mutations. And as more mutations happen, those tumor cells look less and less like a normal cell and more abnormal. And that actually becomes important when we're dealing with immune therapy. Because the challenge with immune therapy is it has to do something to help the immune system figure out, well, how do I know what's tumor and how do I know what's normal? And the more the tumor has differences from normal, the easier it is for the immune system to recognize it and attack. So we'll talk a little bit about that, but that's the PD-L1 story. But so there's this whole more simple tumors, we've got pill drug options that can work really well. The more complex tumors, often that's where the immune therapy works a little bit better. And so Now, instead of just when someone's diagnosed with metastatic disease, trying to figure out, okay, what's the right chemo, I still have to think about that, but I also have to figure out, is this going to be a tumor that is going to respond well to molecular therapy, or is this a tumor that might respond better to immune therapy, and how do we then bring all of that together? And it looks like Danielle wants to interrupt me, ask a question. So I'll let her do that.
I don't want to interrupt you. What I want to do is I want to pause. Because that was a ton of information, right? And I have a lot of questions that I want to ask on behalf of patients. I know Michelle has a question online, but I want to remind everyone in the room and everyone watching that we are doing this for you. So as we're having these conversations, please feel free to interrupt at any time and ask a question. Even if your question isn't about what we're talking about right now, that's why she's here. You all right now have access to Dr. Wakelee. Please take advantage of it. With that, I'm going to go to the online question.
So are you familiar with the clinical trial drug pozotinib and the response that exon 20 768 patients are getting?
Okay, so that's a good question. So getting into the, so the question was about some of the newer drugs that are being developed to treat a specific kind of EGFR. And one of the drugs that they mentioned was pozotinib. There's other ones too, which I'll talk about some. So, so I gave you the kind of simple version of the story with EGFR, right? It gets a little bit more complicated. So it turns out that while there are a couple of specific gene mutations in EGFR that were the ones that kind of unlocked that initial story, and that was something called deletion 19. You heard a couple people mention that. And then there's something called EGFR. It turns out that there are other mutations that also can happen, and it took a little longer to find those. One of them is something called, when we talk about genes, we talk about introns and exons, and we label the different exons that make up a gene, which then turn into the protein. And so there's a specific region of exons within EGFR, the main ones that get mutated. And one of them is in exon 20, and there are some different mutations that happen there. And the exon 20 mutations tend to happen in the same types of people who get lung cancer who get the other kinds. And in some ways behave similarly, but the main difference is they don't respond to the EGFR drugs that you've all been talking about. And so they've been an extra challenge. And the great news is just in the last couple years, there are finally a couple of medications that look like they actually seem to be working in EGFR exon 20. They're still pretty early in trials. And so one of them is this drug called pozotinib, which was the question. There's also one, doesn't have as fancy of a name yet, not the one I was, but it's TAK-788. And we're doing research on that. UCSF and UC Davis have the other one. And so there are trials that are open for people now who have the EGFR exon 20, and that's been really exciting because that had been a really big challenge for quite a long time. And I think it highlights too that the more you know you don't know, and the more you know that there are challenges that lie ahead. But it also means that we're sort of gradually adding in more and more options for people. I think a little bit more on the EGFR story that opens up is with these medications, when we know the exact gene that's the mutation that's the problem that caused the tumor, and we have a medication that works really well to treat it, and yet we're not yet talking about cure, right, when we're dealing with metastatic disease. And that's because cancer's kind of clever. And we talked about how there's a mutation that happens that leads to the cancer initially. And then I talked about how in order for cancers to start to grow in other organs, they have to do other mutations. Well, what's happening is as the cancer cells are dividing, there's constantly new mutations developing. And sometimes those mutations don't give any advantage, and sometimes they do. And when they do, they allow growth in other organs, or sometimes they allow the tumor to then be resistant to whatever treatment it is. So we've known about chemotherapy resistance a long time. We then get resistance to the EGFR or ALK or BRAF drugs. The cancers figure out a way around it. They change how the drug binds, or they do something, or they change the root mutation. So we heard about EGFR going to ALK, or things like that can happen. With EGFR, the most common thing that happens when someone's on erlotinib, which is the Tarceva, or gefitinib, which is Iressa, or afatinib, is that things change and they develop a new mutation. So instead of just the primary one, there's a second one, and it's called T790M. There are other things that happen too, but that's the most common. And when that happens, it took us a long time to figure out what drugs work there, and now we have this osimertinib drug, the Tagrisso, and that works if you have T790M. And now we're trying to figure out, well, maybe should we use that one first? Should we use it second? Like, how do we sequence that? And so there's a lot of research being done with that. But it also highlights the fact that, well, once the osimertinib, the Tagrisso stops working, so now you've got the primary mutation, you've got T790M, then there are other mutations that develop, and those can be, are still, that's kind of the cutting edge, is what do we do there? And so I don't want to, everyone's sort of on their own journey with this disease, and so I don't want to be too specific, and I certainly don't want to sound anything other than optimistic, because it is really amazing to have so many new treatments, but those remain the challenges. Is what do you do when the tumor gets new mutations that are not the ones that we necessarily have treatments for right now? And that's where chemotherapy can come in and play a big role, and is still an important part of treatment. And as we talked about, maybe immune therapy too, in some cases, but not all cases.
I think what Dr. Wakelee is saying, and I think you're starting to get the idea right now, just how personal your cancer is and how unique and different it is. And that shows us how important it is to get genomic profiling, right?
Exactly, yes.
Initially when you're diagnosed, upon progression, and throughout the cancer journey, because it's most likely going to show up different every time that happens, right?
And I definitely want to talk a little bit about the molecular testing and, looking sort of baseline versus NGS, but I do want to get to Don's question first. And then we're going to need a mic in the back after. I don't know if Bridget's still here.
Dr. Wakelee, and correct me if I'm wrong, but it's my understanding that if you have a driver mutation like EGFR ALK-ROS1, you should exhaust all targeted therapies first before trying immunotherapy. And then with immunotherapy, PD-L1 is a marker. Tumor mutation burden is probably even, from what I heard or understand, it's even a better one. But what you're thinking, because now the combinations of immunotherapy and chemotherapy are being fast-tracked for first-line treatment, and it's my understanding the way it works is that the chemotherapy irritates the cancer tumor so that the immunotherapy can see it easier, even without the PD-L1 expression. And I'm just wondering what your thoughts on that are.
So I'm going to kind of answer your question at the same time I'm going to highlight what Bonnie said. It really is very individual as to what's going to be the right treatment for an individual. There are some truths, right? And one is that if you identify before starting treatment for someone with metastatic disease that they have an EGFR mutation or an ALK translocation, that they're going to do better with a targeted treatment than they are with chemo on average, right? There are always exceptions to that. And that, at least in the setting of EGFR, after first progression, for most people, going to the next EGFR drug is the right thing to do, but not for everybody, okay? And so it's not always about exhausting it. And it's much more complex when we get to the more rare mutations, such as ROS1 or MET. And it's not, in all cases, necessarily that the targeted drug is going to be better than starting with the chemo at that point. Okay, so I don't want to make it too simple, because these are important questions for people to come back and ask their oncologists. But I don't want people to leave here thinking, oh, well, they're doing it wrong in my case, because it might not be, right? There are going to be subtle differences. The immune therapy question is a good one, and I was going to get to that, but I'll address it now because it's so critical. We talked about the fact that as a tumor has more mutations, it makes it more different than normal tissue, and therefore the immune drugs can better recognize it. And a way of kind of measuring that is the tumor mutation burden. It's the total number of mutations in a tumor. But the immune drugs that we have today, the drugs like pembrolizumab, also called Keytruda, and nivolumab, also called Opdivo, and atezolizumab, also called Tecentriq, and durvalumab, which is also called Imfinzi. Everything has multiple names. Those drugs all target something called PD-1 or PD-L1, which is a protein that's on cancer cells and also normal cells. And the way that they work, when we're talking about immune therapy, well, they basically block that drug, the protein interaction between PD-1 and PD-L1. And when that is interrupted, the immune cells, it takes away one of the stop signals. So immune cells are constantly having to go back and forth between a pro and a con signal, right? So that we don't end up having our immune system attack us. it has to be very focused, so there's a very tight regulation. And the PD-1, PD-L1 is one of those stop signals. And if you take it away, then the immune cell is better able to come in and recognize a tumor, right? And especially if that tumor is using that PD-L1 to mask itself and look normal. Did I lose everybody? Are we okay? All right, let me explain it one more time. So you got, with an immune cell, when it's kind of surveilling what's going on, it's looking for things that tell it to either attack or not attack. It's also been primed to recognize certain, I don't know, it's called epitopes, but like a little piece of a tumor, right, or a little piece of a cell. And if it sees that signal, it's going to go into attack unless there's a reason it shouldn't, which is a stop signal, okay? And PD-1, PD-L1 is a really key stop signal. So the way that these immune drugs work is if your immune cell's about to go in and it sees the stop signal on a tumor, it says, oh, never mind, and it backs off. But if you remove the stop signal, then it can attack really well, okay? And so if a tumor is using PD-L1 as its major stop signal, and there's a lot of that PD-L1, then of course it's more dependent on that, so when you remove it, the immune drugs work better, right? So high PD-L1 on a tumor is a marker that tumor is going to be sensitive when you remove the PD-L1, you've taken away its stop signal, and it gets attacked, okay? But it's not that simple either. It's much more complex. So you have to have the tumor cell look different than normal, and that PD-L1 signal is important, but it's not the whole story. You can have low PD-L1 and still respond, okay? Where it gets even more complicated is that when you have EGFR, ALK, or ROS, or one of those driver mutations where it's otherwise a kind of a simple tumor, they can have high PD-L1, but when you block it, it doesn't actually have the immune response you might expect because it's not different enough, right? So you kind of need both of those things to have the best immune responses. So what we know from trials for patients with EGFR and ALK is that the likelihood of having a response to an immune drug is much lower than if you have one of those more complex tumors, right? So going back to something we talked about before. And so we know that even though the immune drugs, there's lots of news about them and press, everyone's really excited about them, we know that they're not the best thing early on for most patients with EGFR, ALK, or some of the other driver mutations. And we know that from trials, and we also know that kind of theoretically for that simple tumor reason. And so to get to your question, you were saying, well, you shouldn't give immune drug before targeted treatment, absolutely, but you also probably shouldn't give immune drug before chemo. And the question we're trying to answer is, well, should you give immune drug with chemo the way we're doing for a lot of other patients now? And we don't know the full answer to that question yet. Because with the combination of chemotherapy plus immune drugs, there was just another FDA approval yesterday, Bonnie was alluding to that, that was specifically not if you had EGFR ALK, right? So that approval of carboplatin, pemetrexed, and pembrolizumab was specifically first line for people who didn't have EGFR ALK. And so what about people with EGFR and ALK, and they're ready to go on to chemo? Well, should they get it with immune drug or not? We don't know. And so there are trials being formed. There's been one of the trials that was with an immune drug plus chemo did include that. That was carboplatin, Taxol, and bevacizumab, and atezolizumab. And I'm not trying to lose you all. I'm just trying to explain the complexity of it. It's a complexity. I need you all to think I'm smart, right? So, right, okay. And so in that study, there was a signal that maybe for the EGFR patients who were on that study, that was helpful to have everything. But should we give that 4-drug, or can we do the 3-drug, and how much does the bevacizumab matter? And so those are the trials that are in design right now. And a lot of people think they know and are leaping ahead and doing different things, but we still don't. Mm-hmm. There are things that we think we know that we realize we're wrong about when we do trials. And so that's why it's so critical for us to do them, because we're not always able to guess the answers. And I've been doing this long enough to have enough personal experience with the studies that we were definitely going to have change everything that were negative. And so a little bit cautious there.
But what Dr. Wakelee is saying, is what we said just a few minutes ago about how unique you all are. As we all have a fingerprint, which is very unique to who we are, we have a body print that is also very unique to who we are. But what I can tell you, and I'm obviously not a physician or a researcher, but more therapies have been approved for lung cancer in the last 4 years than in the last 5 decades. Mm-hmm. So what Dr. Wakelee is talking about is this thing is moving at rapid speed. You know, so hope resounds in everything Dr. Wakelee is saying. I mean, it's like rocking and rolling.
I have a question based on something, since we're on this sort of immunotherapy train, and then I think you have the mic for your next question, yes. And that is looking at, you know, kind of what you were saying earlier about EGFR and where we thought those TKIs were who they were and weren't going to work for originally. What does that look like in immunotherapy, specifically as it relates to Nancy, who has small cell, because she mentioned that in some of the recent information coming out about that. Can you talk about that?
Right. So with small cell, there's been a lot of research as well. Now, a lot of this information about the specific mutations. With small cell, it's a bit more complex as far as, but it also means that small cell tumors in general have more mutations, and therefore, theoretically, and now we know in reality, are going to respond better to a lot of the immune drugs. And so what Danielle's alluding to is there was an approval just last week for immune therapy specifically for small cell. So it's one of the drugs that we've had, one of the PD-1 drugs, but now we can definitely use it in small cell in the right setting. And there was also a press release that for some people, combining immune therapy plus chemo in small cell might be better when you're just starting off on treatment. And that's all. We haven't seen all the data from that, just sort of the press releases. So just like in non-small cell, in small cell now, it's like every month there's something new happening. And so that's been really exciting.
Which is huge, because it's the first time a new approval in the last 20 years or something for small cell, right? So that's a huge, huge, huge step and milestone in this space. Right. Okay, on your mark, as I go.
Thank you. Yeah, a lot of discussion on EGFR, and about half of the people said that that was recognized there. But they always say EGFR like that means something. And how many different kinds or how many different mutations are found in EGFR that causes cancer? And is there a progression where it normally starts with a particular exon and then progresses to mutate others, or does it kind of stay the same mutation for your entire life?
That's a great question. So it turns out that with EGFR lung cancer, it's very interesting because there are two main mutations that are found. And that's the vast majority of EGFR lung cancer has either exon 19, so that's exon 19 and then it's deleted, so part of that's removed, or a specific mutation in exon 21, which is called L858R. And so when someone's diagnosed with EGFR lung cancer, the vast majority of the time it's one of those two. Now, we talked about one of the other ones, that exon 20, and then there's also a couple we see in exon 18. And their response to the EGFR drugs is a little bit variable, and we don't know as much about them because they're rare. Usually, once you have a primary EGFR mutation that's the driver, the one that really started the whole thing off, that never goes away. Other things can add to it, and they're oftentimes, so sometimes we get that other EGFR mutation I mentioned, the T790M. However, there are other genes then that become mutated. So that's what ends up happening, is it starts in EGFR, but then other genes also mutate over time because those cancer cells are dividing more and have more opportunity for mutations.
How many mutations are there normally before you kind of declare cancer? Is a single mutation, or is it 4, 5, 6 mutations before it actually becomes cancer?
So it can be a single mutation. It gets again, I don't know how much complexity to put into this. So we know that like when we have a model like an EGFR where there's a single mutation that we know was kind of the driver, that's what started the whole thing off, that's then there in every cancer cell. But over time, different pockets develop, right? So the cancer cells that are growing in the liver are going to be, they still have EGFR, but they're a little different maybe than the cancer cells that are growing in bone, even though they still have EGFR, which are a little different than the cancer cells growing in the brain. And we don't understand why it grows in different organs very well yet. That's an area of research. We also know that when a mutation develops that gives it a growth advantage, those cells can then start spreading to other places. So they might start off in one place, but then they pick up and grow in other places too. So the longer someone's living with cancer, on average, not always, and certainly not applying to people here, the more sort of subclones there are. A clone being where one cell then gives rise to more. Does that make sense? So it's you kind of get that one main one, and that's always there, but then other things can add on to it.
And I think this points back to the importance of, and we haven't even gotten into tissue biopsy versus blood biopsies and all that kind of stuff, but why you need to be having these conversations with your oncologist, and why at progression, rebiopsy might be a good idea. So maybe you can kind of talk a little bit about that. And then Barbara got you, yeah.
Okay, all right. So thanks, Danielle. So, right, so I'm talking about all these mutations, but it's not like someone has cancer and they walk in with a sign that says, I have this mutation in my tumor. We have to figure it out. And I mentioned when we were first learning about EGFR, they had to what we call sequence. They looked at every single base pair of the gene to figure out, well, what was different here versus there. And once they saw this pattern, then there's shortcuts of how you can test the tumor to look for those specific genes that we need to look for and specific mutations we need to look for. And so for a long time, when we tested for EGFR, we just looked for those two main mutations, so we missed the other 10%. And then when we started recognizing these other genes are really important, we had to figure out, well, how do we look at all of those? And there are different kind of ways of doing it from the tissue. You've heard people talk about next-gen sequencing or NGS, and that's where you look at several hundred genes, but focusing in on the key areas where mutations are more frequent. And by doing that, you don't have to sequence the entire genome. You can just focus in on a few things, but the key ones. And so there are now a lot of companies that are able to do that on tissue and do it fairly quickly. And most of the universities have their own way of doing it. Everybody thinks theirs is better, right? But they're all looking at that same thing and really emphasizing you just have to look at enough genes. Because you don't know. I mean, I just today got to tell a patient about a MET exon 14 mutation. I mean, she was not someone who was going to be able to tolerate chemotherapy well. We didn't know that we had any options, and then we were able to get this test. Now, in her case, it wasn't from the tissue because she didn't have enough. It came from blood. And so that's sort of the next frontier that we're working on is how do you do this testing that you should be doing on the tissue with the tumor from blood? You're like, well, wait a second. You just told me the tumor, the thing that makes it different is those gene mutations, and that's different from the rest of the body. So if I draw my blood, which is normal tissue, why is that going to tell me what's going on in the tumor? I mean, that should be the question. It turns out that when a tumor is dividing, it's also dying, right? There are parts of it dying, and they're going to put little pieces of DNA into the blood. And that happens to all, there are little pieces of DNA in all of our blood from normal cells as well. But when the tumor's doing that, it's going to be those little bits of DNA are coming out that most of it looks like everything else, but there are little bits of it that have those mutations. So if you know what mutations to look for, you can test to see, well, do I find that gene mutation in the blood of the patient? Well, then I know it's coming from the tumor because that wouldn't be there normally. And so that was the start of this. Now we're getting to a point with research, and again, Stanford and other places, where even if you don't know someone has cancer, we're getting to a point where eventually, we're not there yet, you might be able to do testing on the blood to see, well, is there even cancer to begin with? We're not there yet, but we're getting there. Or after someone's had a surgery, can we figure out if there's still any cancer DNA in the body to then look differently, watch for surveillance, or give treatment to try to prevent the cancer from coming back? Or right now what we can do is if someone's had a blood draw and we know that they've got that primary EGFR mutation, and then their tumor's starting to grow, and we know they do another blood draw, and now they've got that other EGFR mutation, then we know that we could treat them for that other mutation, or maybe a new mutation is developed. And so the blood draw technology, the circulating tumor DNA technology, is allowing us to really, without having to biopsy, for some patients, track what's happening with the tumor over time. It's not perfect. And sometimes we still have to do biopsy, which is what you're talking about, and repeat biopsies can be very helpful, particularly when there's a driver mutation. Now, if someone's tumor has a known mutation that's not one, or a lot of mutations that's not one that has one of these pill drugs that we know about, repeat biopsies are probably going to be a little less useful because we're not going to change treatment based on that. I mean, because they're going to be focusing on immune therapy, we're going to be focusing on chemotherapy. There's some utility to biopsy sometimes for the immune therapy, but not always for chemo. We still don't have great ways of picking best treatment based on that. So for some people, especially if there's EGFR, biopsy at the beginning, biopsy later, or blood biopsy, if you can get the answer. But for sometimes it's not right. So again, getting back to that individualization,
right.
The message of testing when someone's first diagnosed is so critical. Still happens, people come in for second opinion and they don't know what the mutation is in their tumor.
Right.
And then I'm having to wait until I know that to treat. And half the time or so, I get information that helps me figure out something else. And half the time I get information that says, no actionable tumor. But that doesn't mean we don't have treatment options. We still have a lot of options. We just know what's going to be the better one based on that information. Does that make sense to you? Okay.
And our blood-based biopsies, we're hopeful that at some point in the future we can use those to chronically manage cancer and actually see progression in the blood before you see it as a tumor on the lung. And look how much sooner we're going to be able to start treating a patient rather than waiting until you get another CT scan and you see a tumor, because we know it's circulating. Right.
Then the question becomes sort of how soon is too soon, because the drug might still be working in the majority,
Exactly,
right?
and that's something, and that's then managing the anxiety around the disease too. Sometimes having that information, just like sometimes seeing something starting to grow in a scan. Well, are we at the point of switching yet or not? And sometimes yes, and sometimes no.
I want to get to Barb. She's being so patient.
Okay.
So I think we've all drank the genomic profiling Kool-Aid.
Right.
Because at least any of those of us that have been here for a while, and when Dave was diagnosed, of course they did that, and that's how we found out he was EGFR, exon 19. And then when we started to see some progression, they did a blood biopsy that showed not enough anything to tell us anything. So then they did a tissue biopsy of the lymph node, which was what had started to progress. And we just got that report today after 7 weeks, which I won't go into that. But it says there's no information on the tumor mutational burden. There's no information on the MSI. So kind of what are the limitations of this stuff, and what do you do when you're not getting any answers from that?
Well, again, to reemphasize what I said, I mean, not every tumor is going to have a mutation that's going to give us a new treatment option. And not every biopsy is going to give us new information that's going to let us help. So some people from the very beginning, their tumor doesn't have one of those that allows us to give a different treatment choice. And for someone who does, it might evolve over time in a direction that also doesn't allow us to change, right? And so that's the frustration, because I think with every scan that we get, every blood draw, every tumor testing, you want it to be meaningful, right? To be able to allow to inform improvements in what's being done, or to say the cancer's gone, right? One of those two things. And so it's frustrating when results come up that don't allow us to change treatment, but that's often what happens, right? And it just says, well, if we, in a case where someone's on erlotinib and we're looking for that new EGFR mutation with the T790M to allow a switch to osimertinib, when we don't get that, it seems like, well, test didn't work, failed test, but oftentimes it just means that that test, that's not what happened. Now, there are a lot of companies that are doing this testing, and they're not all the same, so that's also part of it. I mean, you mentioned, and it's one of the ones that really does a very good job. But we have to be careful, because I do get frustrated sometimes when a patient has had a tissue biopsy and they come in and these are my results, and but it didn't look at these genes, right? And so trying to get that word out that there are more and more genes which are actionable, I think that's critical. Now, the tumor mutation burden, that's come up a few times. Again, it's a marker of how many mutations are there, but it's not a validated test yet in the same way. I mean, it is in with one particular assay, with one particular company on one particular trial, yes. That was a clear answer. But in general population sense, we're not looking at tumor mutation burden uniformly, and we still don't know exactly what it means. Right? I mean, we know if someone has really high PD-L1 and really high tumor mutation burden, that is a person whose tumor is really likely to respond to immune therapy. But when you have discordant results, we don't know exactly what that means. I could quote all the trial data, but we still don't really know what that means. And we also know that high PD-L1 doesn't always mean response, especially in the setting where there are mutations, right? So it's more complex with immune therapy, and it's more complex with kind of figuring out, well, the evolution of the tumor over time. Sometimes it's helpful and sometimes it's not. So
I think, I'm sorry.
Yeah.
Is it true that that's why they're doing clinical trials with immunotherapy and chemotherapy to see if the chemo will give the immunotherapy a boost?
Well, what we know with, so people who don't have EGFR ALK, we know that combining chemotherapy and immune therapy, first line when someone's just diagnosed, just getting started, looks better than chemotherapy alone for most people, right? There are always exceptions. And we don't know exactly how to bring that in for people with EGFR and ALK, though we do know that if there's EGFR, and I'm using that, it's obviously the list is a little bit longer, but for shortness, that if they have that it's still better to start with targeted drugs. So those are kind of the truths that we know, and there's a lot of nuances to that we are still trying to understand. I do want to branch a little bit into earlier stages of cancer because we haven't talked about that yet. It's kind of started to hint at it a little bit. Most of what I've talked about is for people who have metastatic cancer. But there are a lot of people who are found with the cancer at earlier stages of disease, stage 1, stage 2, stage 3, and you know, I'm not going to quiz you on all the staging, but stage 1 basically means it's just the tumor. It hasn't figured out how to grow anywhere else. And stage 2 means it's the tumor, but it figured out how to grow in lymph nodes in the lung. And then stage 3 is it figured out how to grow in lymph nodes that are in the center of the chest. And then stage 4 means it figured out how to grow anywhere else. So stage 4, obviously, there's a lot of variability in what that means, okay? But it just means it figured out how to grow somewhere else. And the treatments for stage 1 and 2, for most people, we try to remove it with surgery. And some people get focused radiation, and that can be great. And then we will try to improve cure rates by adding other treatments after, whether that's radiation, rarely, or chemotherapy a lot of the time. And what we're trying to figure out is, well, should we add these targeted drugs if someone has an EGFR mutation or ALK that's found early and we can remove it, and now what do we do? And there's a lot of research there. We don't know yet what's the answer. And we're also trying to figure out, well, should we give immune therapy and the chemo or after the chemo? And that's also research that's being done, and we don't know. And then with stage 3, it's really very, very complicated because sometimes it's surgery and sometimes it's not, and sometimes it's radiation and sometimes it's not, and most of the time it's chemo and sometimes it's not. And maybe we should do immune therapy and maybe we should do targeted treatment, but we still don't know. And that's where there's a lot of clinical trial work so we can help know for the future what's the best thing to help cure people, right? And then there's what we know about metastatic disease and what we know about early stage, and sort of how do we bring them together, because it's not always, you can't always take the knowledge from here and bring it into here.
So I want to just, I have a comment.
Okay.
When we're talking, and then I see you, Ron, talking a little bit about the genomic profiling, and I think one of the things that's, and this is kind of to tag on to what Barb just said about some of the frustrations around not enough of this, that, or the other to gauge whether or not there's this, that, or the other. I know there's other folks in the room who have had the same challenges, and although I believe in, obviously, we believe in patients being educated and informed, there's a challenge with some of the commercial diagnostic companies out there in the way that they are communicating this back to the patient. Well, I know it's generating confusion in a lot of rural sort of community oncology settings, let alone when you're handing a report that's this thick to a patient who doesn't know what they're looking at. And so it's one of the reasons although we encourage this testing be done, and of course for everyone, we also highly, highly, highly encourage that conversation happens with your physician as soon as you can make it happen, or somebody on your care team,
right?
Because it's insanely confusing.
Well, and it is confusing because now a lot of the reports come back and these are the genes that are mutated, but then they take it a step further. And they say, oh, and these are all the treatments that you should be thinking about, and often goes way off into tangents. I'm like, why are they recommending that? And I can say that as someone who, my job is to really know what's going on the cutting edge with the research and what all has been reported. But if I was in the community in practice and I was treating 10 other diseases, I'm just like, oh, okay, they said to do this, maybe I'll do that, right? So if you're in that setting and you're not really sure, you get a sense from that your oncologist doesn't quite know how to interpret that, not because they're not super smart, but because it's just changing so much all the time. It's okay to say, hey, you could, do you want to have me get another opinion, or do you want to just call up your colleague? I mean, again, part of my job is to educate not just my patients and help them, but to help people in the community who don't get to focus just in one disease. And we take calls like that all the time, or emails back and forth. And I think that's really helpful because I'll get those too. And there are things that we learn all the time also with the really rare mutations. But having a treatment recommendation is sometimes helpful, but oftentimes more confusing than not.
Right. Ron, I see you.
Thank you very much for the information. I mean, it's a fire hose.
Sorry.
Fire hose.
I had two questions I'd like to see if you could address, or issues if you could address them. One relates to what's the current state of research or therapies for someone with KRAS mutation? Because we haven't talked a lot about that. We talked about EGFR and ALK and ROS1. KRAS, I think, would be of interest to some of the people. The other one is there research on the outers? Why are some EGFRs lasting so long? And others, they're also the same exon 19s. Maybe they're not type 1s. I don't know. Why is it that some of these outers are surviving so long? Is there a longevity study that tries to figure that out?
And I want to tag on to that question. So KRAS, EGFR, and what's working and what's not. And I lost it. No, because is there a difference in somebody like Tina who had chemo up front and then she went on her targeted therapy? And then is there a difference in using a Tarceva versus an Iressa versus an afatinib, versus how does that play a role? And how do you decide which drug?
So to get back to KRAS, to mention a little bit, it's definitely one of the mutations that we know to look for. It's the most common driver mutation in adenocarcinoma. If you look across the board, but because of the complexity of all of the genes that interact with it, it's been one that's been very hard to treat. So we were really excited a few years ago about something called MEK inhibitors and combining those with chemo, and then they did help some people, but not enough. And so there's ongoing research now, and I mean, we actually just opened a trial last week that's combining a couple of oral medications, and there's ongoing studies at other institutions where we keep trying to push that envelope. It turns out that KRAS also, because sometimes, though not always, there are other mutations along with it, tends to respond better to some of the immune therapies than some of the other driver mutations, right? So it's not that it's nothing that we can do, it's just that it doesn't have that pill drug option. It has chemo and immune therapy and maybe some pill drugs, but we're still trying to work out what those should be. And did that answer the KRAS part of it enough? Okay. And then your other question, I was really focused on outliers, right? Who are these outliers? Why do some people do phenomenally well, and why can't we have everybody else be like that too? And that's a great question, and it is one of interest. And we're trying to figure out, well, is it something to do with the other mutations that go along with EGFR or ALK or whatever it is that that person has? Or is it other things? And the way I look at it is those tumors, right, over time, they're all changing in different ways. That's why they become resistant. And I don't think we've really figured out well enough how to predict which direction it's going to take. And that is something people are trying to understand better, but we just haven't really figured it out as well. And then, Danielle, you were asking about some of the other EGFR drugs. And so there are multiple EGFR drugs available. When we kind of group them into the first generation and the second generation and the third. And that has more to do with what EGFR mutations are getting. So the third are getting the T790M in addition to the others, and also less of wild-type EGFR, so they have different toxicities. And the second generation gets that T790M a little bit, but also gets some wild type and some of the those two driver mutations we talked about. And when people have tried to do head-to-head trials, there have been a couple of those. One is a drug no one's ever heard of called dacomitinib, head-to-head with the gefitinib Iressa. And that looked better, but more toxic and not approved yet, and maybe will and maybe won't, we don't know. And then there was a head-to-head of the osimertinib, which is the Tagrisso versus the erlotinib Tarceva. And that one, on average, was looking like maybe it worked a little longer, except if someone's been on erlotinib for a while and then goes on to the osimertinib, well, then they would do as well as if you just started with it. But then who are those people where they're on the erlotinib long enough that they would have been much better starting that way. Obviously, a lot of people in this room have done really well with that approach, right? So there's a lot of different questions within that question, and it gets back to Bonnie's point. It's all individual, and there's some of it we understand, and a lot of it we still don't.
And there's markers we don't know about yet. If you look at a company like Loxo Oncology out in New York, they created a drug for a marker, an NTRK marker, and we didn't have that 6 months ago or 7 months ago. So it could be that Tina, who is an outlier and doing so well on this one drug for years and years and years and years, this is where data comes in. This is why we need research. This is why all of you in the room are contributing to research, whether you realize it or not, because you're giving your specimens every time you have some kind of an event, whether it be a biopsy or whether tissue or blood. And that data is being accrued. And that's where we're going to find, it's like, oh, look at this. This group of patients all started with an EGFR mutation, blah, blah, blah. But look at this down here. This is crazy. There's like 30 of them that have this marker that's known in other diseases, other cancers, whatever. This is something we need to study. This could be the next therapy.
I think one of the things that's kind of standing out to me tonight, and it does most living room nights, and particularly with a lot of new folks in the room, hopefully a ton of new folks online, is the importance of being with a physician who specializes in this disease, because it is so very complex. Each individual subset is insanely complex, and this is, when you in the community setting, and where Leah spoke a little bit earlier about our community Centers of Excellence program, most patients diagnosed, 80% of them don't have the benefit of a Dr. Wakelee, right? Which is one of the reasons we bring this to you, and we're like, you've got her for 2 hours, ask your questions, right? They don't have the benefit of it, and those I feel, even for those physicians, because it's seemingly insurmountable ask of them to know everything there is to know about every cancer that walks in the door, right? Solid tumor and blood cancers. They have to treat what walks in the door. So,
and yet there are still some basic principles, right?
Sure.
And so I think for most people, most of the time, right, as long as some testing's being done and you know the key gene mutations, and a lot of people aren't going to have those. We still have the chemotherapy. We still have now a chemotherapy and immune therapy, and all of this understanding is so critical for helping everybody. But it's still right now, not all folks are necessarily having to get a biopsy every time or having to be with a super specialist, right? I mean, and so I do want to make sure that people who don't necessarily have access don't feel that they're tonight. It's good to have this way to kind of disseminate that information and for people to learn and have ways to ask questions.
Exactly.
And a lot of times just to get reassurance. I mean, a lot of the people that come to me for second opinions can say, you're on the right path.
Yeah.
They're doing the right thing.
Yeah, exactly.
Hi, my name's Tom, and I'm here for my partner who just passed 2 weeks ago.
Yeah.
I just had a few questions. We bumped our heads against testings and 2-week, 3-week waits, PET scans, and this and that. So my question is there a standard for genomic testing for all labs? And question number 2 is, who is overseeing the lab testing, and is it worldwide, or is it this and that and the other thing? And the other question is, who's overseeing to make it equal results and waiting periods for those results? The other one is insurance/PET scans. And then one last question, and these are a lot, but these are things that I've been pondering for quite a while. Every doctor that we saw, and oncologist, or the doctor or the lab that we went to, each one of the doctors wants their own lab to have their own results. They don't want to take all of our information, like when we moved from Wyoming, we moved here, and then we went there, and not only 2 places, but each one wanted their own results from their own lab. It's almost like they did not trust the results from somebody else doing it, And that's why I was back to the wondering if a genomic testing is done here, why wouldn't it be recognized in Australia?
So those are great questions. So I'm going to try to answer that. So there is no uniform testing, right?
Okay.
There's no standard. For certain things, like looking for an exon 19 deletion or an L858R, that's relatively straightforward. I mean, there are ways that we know to do that, and that's pretty reproducible. In order for, and I'm not in this field, right? But I order a lot of tests, so. In order for a test to be available, it has to be reproducible. So you have to show that in your company, whenever you run this assay with this particular sample, you always get the same result. So that reproducibility is really critical, right? Okay. But is it necessary, and for things like an EGFR test, you should be able to then take that same and run it across, right? So that accuracy part. But then with some of the other aspects of which genes you're looking at and how detailed to look at, then there's a lot of infighting as to, well, we're more accurate than this person or that person, or lab, or companies. And so there's a lot that's uncertain. It's different than, say, when there's a drug that's being developed, it has to go through very specific trials, comparisons, and then that drug is made by that company. And if you want to make it generic, you got to go through all sorts of different hoops to make it available. But if it's a test, a testing company, you just have to show every time you do the test, it's reproducible. And again, there are people from pharma here I know who have different, that can give it more detail. But that's sort of the way. And so that's why you have 20 different companies offering similar tests. And how do you know which one's the right one or the best one? That's harder to know. I mean, it should be reproducible.
Yeah, and I understand there's so
many. Yeah.
I understand there's so many tissues that they can take from one biopsy. And then there's multiple biopsies, like we were talking about, that can read other things from it. And I know the tree breaks down huge into many areas of all the different cells. So thank you so much for being here.
Yeah, and then also to your PET scan question, though, and and what's covered. I mean, I'm actually battling for one of my patients right now where he fortunately doesn't look like he's any active disease, but he had a couple nodules, and so I wanted to get a scan at 4 months. He's on surveillance. And his insurance company told me I had to do 6. Yes. And we had to do this peer-to-peer where we're on the phone talking to the doctor who got the job at the insurance company because they didn't get a job anywhere else. And, and it's I know that's taped, sorry to anybody in that industry. But, it's like, it's frustrating because, I'm trying to make a decision about what I think is right for the patient, and yet I'm being told that I'm not following a guideline. And yet, I'm on some of the committees, right, some of the guidelines, right? I mean, that can be really hard. So it's frustrating for us as it's frustrating for the patients. At the same time, there are some doctors who are trying to get a PET scan every 4 weeks for a patient, and that's not good either, right? So we have to have some way of finding the right balance.
The reason I even brought it up was because the ALK came on so fast. 5 weeks later, she's passed.
Yeah, I'm so sorry.
And not that a PET scan would have helped in between or anything else, but by the time we got results from one, she'd already got it so bad that it was it was irreversible.
That's really tough. I think another part of your question was around the timing.
Yeah.
And we try to get test results from molecular testing back within a couple weeks.
Yes.
But it's a very complex test. It's not. I mean, some of our tests like PD-L1, you get a piece of tissue, you slice it under the microscope, you put a stain on it, it's either positive or not, you can get that in a day. Yeah. But for the molecular testing, you have to get the DNA out of the tissue, which sometimes doesn't work well. Yeah. And then you got to run it through a whole bunch of tests, and so it just takes time. And we're pushing to make it faster because I feel that with my patients when we're can't start until we know we can't, and it's so hard.
I understand how many people are in line for these tests. So
yeah.
I was just going to add to some of the commentary here. One. If they're asking for repeat tests depending on location, it may be because they're looking for new information. It may not be a trust issue. So perhaps depending on how much time had lagged, they might be looking for.
There's a lot of things.
Yeah. The other thing that I would also comment, and perhaps some of the industry experts in the room would know, as well as you, Dr. Wakelee, is that 14-day rule still in effect, and is that causing trouble in terms of timing?
That's actually fortunately going away. So what she's referring to is in the past, it used to be if someone was in the hospital, you couldn't order molecular tests until they've been out of the hospital for 2 weeks. I mean, that was ridiculous. That's, I think, gone away now.
Okay. So. Thank you, guys. We've got like 7 minutes left, and
And 15 seconds.
And 15 seconds. So I'm fearful that if we ask one more question, it's going to go like over time. So I'd rather just kind of wrap up here, and then maybe Dr. Wakelee can stay for 5 or 10 minutes and answer any questions that might come after. But I want to make sure to do our sort of due diligence and thank all the appropriate people that need thanking. First and foremost, thank you to Dr. Wakelee for coming out and talking to us tonight. Like I said, this is a meeting that sometimes, well, I used to worry about being able to fill 2 hours, and now I'm always like, man, we need 4, because there are so many great questions that come out and so many other things that we could continue to talk about. And I saw some folks walk in the room late that I'm certain have questions. And some folks that were quiet and here all night that I'm also certain have questions. So thank you for coming out. Thank you to all the patients in the room. Again, I know we started this meeting thanking you all for sharing your stories, but thank you for asking the questions. Like I said, we do this for you. So we're grateful for you sharing your stories and your time and your efforts to be here. Thank you for everyone online. Again, those of you watching tonight live, and those of you who will come back and watch post live. I, of course, want to thank the Office Bar and Grill once again for donating the food for tonight, for pasta night. Like I tell everyone, the third Tuesday of every month is pasta night here. So, so don't have pasta the day before, the day after, the third Tuesday. It's here. Thank you to Peninsula Television. For what happened? What did I miss? Thank you to Penn TV, Peninsula Television, for those of you in the room who are new. They're local cable who come out here every month, the third Tuesday of the month, way before this production starts, and they set up all this great equipment. But not only do they do that, then they play it over and over and over all month long on Channel 26. So
if you can't sleep at night,
you're probably certain to be able to find it on Channel 26. And we joke that we have been out places where people have randomly walked up and they're like, "You guys are the cancer ladies on TV, right?" And we're like,
on
"Yeah,
TV.
yeah." So thank you to Peninsula Television. I have to thank our sponsors, with which there is a very long list, so I need my glasses and my paper. These are in no particular order. AbbVie, Amgen, AstraZeneca, Boehringer Ingelheim, Bristol Myers Squibb, Celgene, Genentech, Lilly, Medtronic, Merck, Novartis, Takeda, and the Yahoo Employee Foundation. Good. Without their support, we could not bring this to you guys. So huge, huge, huge thank you to them for believing in what we're doing here and recognizing the need. Do you have anything to say upon closing?
The only thing I have to say is, this happens once a month, come hell or high water. But we're on the road. We're doing remote living rooms regional. All over the United States. You just did one in. You did 2.
Florida and Denver.
Florida and Denver, Ross Camage, so we. People are begging, me too, me too, me too, me too.
And
so
we're coming
education
up next
is huge.
with some of the regional living rooms in Arizona and Los Angeles, so be on the lookout for that. But I also want to hand it to Katie, because we've got a couple minutes left to talk about. There she is.
Hi, everybody. My name's Katie Wilcox. I'm the
newly engaged this last weekend. Yay!
I know.
Yay!
Thank you. So I'm the Senior Manager of National Events, and I wanted to share some events that you all could participate in, and you all from home. So we have 2 5Ks, a ton of 5Ks coming up, but the 2 I'm going to mention are the 2 coming up most recent. So we have a Hollywood 5K in Florida. That's on September 16th at Charnel Park. And I hope I have this right, it's our 5th annual. So we're going back to that community. We're partnered up with some amazing hospitals. So if you're in the Hollywood, Florida area, please come out and join us. And then we have our 10th annual San Francisco, I always want to call it a 5K, but it's a loop. It's more than a 5K. Come out and please, please participate in that. It's the 10-year anniversary. If you don't have a team, join Bonnie's team. Bonnie, we can give you the link, and Bonnie will happily give you that link. But there's going to be so many. Say it again. and it's Saturday.
And
so
Ron's
that's
going
on.
to wear a red dress.
Yes.
Right.
And you guys, so that's Saturday, September 29th, 10-year anniversary, and it's a superhero theme. So we're doing our amazing marketing person, she was right here, but she came up with it's superhero survivors and superhero studies. So we're going to be sharing all about that the day of the walk. Please come out and join us. And then really quickly, gala event is coming up. We're looking for nominations for star caregivers. Please send those nominations in to Jennifer Hughes. If you're interested, I'll give you her email address. And then lastly, because I never do a plug for this, we have an endurance program. So if you know any long distance runners or people that are just loving to do the running, find me. We have bibs to some amazing events like the New York Marathon, where they can participate and fundraise for the foundation. So that's it. Thank you.
Thanks, Katie.
Our next living room is on September 18th, which is my daughter's birthday, which neither here nor there.
And who will be here?
Dr. David Gandera from UC Davis, who Dr. Wakelee talked about a little
Sometime
bit
we're going to
tonight.
get both of you here at the same time. That'll be awesome.
Is going to be here, and he's going to talk about making sense of immunotherapy. Dr. Wakelee touched on it a little bit tonight, and we've touched on it in some recent living rooms. There's some stuff we know, but still I think a lot of things we don't know. Right. So I think he's going to touch a lot on maybe what we don't know, but where he thinks we might be going based on some of the studies and trials that are being done. So please join us on the 18th. Big thank you to Sally for bringing dessert, but I also want to give her the floor for the last minute because she has a couple of announcements to make. You need a mic, though. Oh, boy.
Oh,
geez.
boy. Who took your mic?
Jeez. There you go.
Hi, Tina and I have organized luncheons the last few years, and people at the last one in, I think it was February or March, whenever it was, said they wanted them more often. And these are not part of the foundation, although Bonnie and some of the people that work here do come. I don't know for sure where it's going to be yet, but I coordinated with Bonnie for a date that she'd be able to attend. So that's what this was, and I don't know if it went around to everybody, but it's just a chance for us to get together away from this setting.
And we talk about sex and all kinds of
Yeah.
things. We don't talk
Wow.
all the stuff we can't
Wow.
put on TV. And the other
Wow.
thing
Still
is,
live.
I've also organized.
Still live.
Again, these are called this is called a grassroots fundraiser, but Amici's Pizza in Redwood Shores is going to give us 25% of everything that's spent on Giving Tuesday, which is Tuesday, November 27th. It's for dine-in or take-out, and they'll be open from 11:30 in the morning till 9 at night, so you can go for lunch or a snack or for dinner. You will have to show them the flyer, which we will put out and mail to people. But I just want to start talking about this now.
That's
and,
awesome, Sally.
They said food or drink, but I never got an answer whether they were going to come do that for the wine
And that's for
beer.
delivery at home, too?
Yeah, it
Awesome.
says dine-in or take-out.
Awesome.
And so it's the 27th of November. It's the Tuesday after Thanksgiving.
I promise you will get reminders about it.
Oh, yeah.
Right. Sally is relentless.
Yeah, I am relentless. That's my reputation. But anyway, so you'll get information about this too. But if you are interested in the lunch, you're not committing to it. You're just giving me information so I can get information to you.
Thanks, Sally.
Deborah, you have to be there.
Thanks. Thank you, everybody. Please come test out the coffee crunch cake. I think it's from Deanna's, so I'm pretty sure it's
No.
amazing.
It's from the cakery in Burlingame. Usually I go to the city, to Japantown, to the Sweet Stop, but they were on vacation, so I'd heard about this one. So we're trying
Anyway, please come
it
get
looks
some
like
cake.
Tony's talking
Thank-
about
Shoot, we can stop filming now, but what I need first is everyone to back up under the hope sign because James is going to take it. James didn't introduce himself, but he's our videographer, photographer extraordinaire. So please come line up in front of the hope. Thank you, everybody.
It'll be hung in the post office.
I don't know where to put it, right?
You offer them cake and then you make them work for it.